Okay, most of you already know that proteins are made up of amino acids. And amino acids, there are 20 of them. Some are essential, meaning that you've got to intake them. Your body cannot make them. Not all of them, but some of them are essential. And this is the general format of amino acids. They've got a carboxylic acid on one side. And generally, when you write an amino acid, you usually write the acid side to the right, the amino side to the left. Notice in its native state at a physiological pH 704, the amine is an ammonium cation, and the carboxylic acid is an anion. Okay, and then you've got one carbon in the middle that has the ability to have different functional groups hanging off of it. Now, they call it an alpha carbon. That alpha carbon happens to be a chiral carbon also, meaning it can have a D and an L form. Now, remember from carbohydrates, the native form of a carb is the D form. With amino acids, it's the L form. Now, there are four basic groups of functional groups on the amino acids. There's two neutrals, one's polar and one's nonpolar. Then there's a basic one and an acidic one. This is a really important amino acid, cysteine, because of that thiol group hanging off of that alpha carbon. Now, that thiol group can bond with heavy metals. That's the issue. It also can form a covalent bond in terms of cross-linking. It's a very important amino acid. We're proposing to do a undergraduate research project using cysteine to monitor arsenic in irrigation water up in the rice fields. And we're coordinating with the Colusa High School there as well, their science department. So it should be really interesting. Anyway, so what we're doing is using cysteine to grab hold of that arsenic. And then we analyze through atomic absorption how much arsenic is there. Okay, so this is what the neutral, non-ionic side chains look like that are non-polar. Notice most of them are alkanes. By the way, let's look at this one here. Glycine is the simplest one because the functional group is just an H. Now remember, chiral has to have 4 independent groups hanging off of that carbon. So if we look at alanine, for example, we have, those are the 4 groups coming off the alpha carbon, and they're all different. So that means that carbon is chiral. So the alanine can be either D or L. Notice with glycine, though. Glycine only has 3 different independent groups, not 4. So glycine does not come as a D or an L. It's a non-chiral carbon. It's the only one. Okay, so the neutral, nonpolar are all basically alkanes. This one's a little bit different because of that S there, but it's still pretty nonpolar. Neutral, non-ionic, neutral, but polar basically has got hydrogen bonding going on. And then there's this right here. All those display strong IMF. Most of them are hydrogen bonded. So very polar, but non-ionic though. The basic ones are all ionic, strong IMF. Okay, and they all involve nitrogen compounds. And then lastly, we have the two acidic ones, and they've got a carboxylic group at the end. Now, glutamate is another interesting amino acid. It's used as a flavoring agent in cooking. So when you eat mushrooms, for example, it's got a lot of glutamic acid in it, or glutamate. That's what gives it an umami flavor. And there's a lot of things you can add to your cooking that have this in it. This is what MSG is made out of, monosodium glutamate. So right here would be a sodium. So this would be MSG. Now, there's a big controversy about MSG because the natural form of glutamic acid or glutamate is the L form. When you buy MSG at the store, you don't buy the L form, you buy the D and the L form mixture. And some people are sensitive to the D form of glutamate or glutamic acid. Some people are, some people aren't. Like I cook with MSG a lot. I have a very poor sense of taste, so I have to enhance all my flavorings a lot. So I use MSG a lot, and I'm fine with it. I don't get sensitive to it. Okay, so I already mentioned the alpha carbon is chiral. So that means everything can exist as a D and L except glycine. They call it an achiral amino acid. Another thing interesting about amino acids, they can be both a acid and a base at the same time. And they can be ionic at the same time as well. And it's called a Zwitter ion. A Zwitter ion. So whenever you see an amino acid, it should be written like this, not like this. That does not exist. Now, there's another phenomenon with amino acids, polypeptides, and proteins, and that's something called the isoelectric point. That is very important, very important. The reason why it's important is at the isoelectric point, that protein or polypeptide is non-ionic. Non-ionic. Now, most of the time, the pH for a protein is not at its isoelectric point. It's above it or below it. If it's above it, then the protein or polypeptide has a negative charge. So let's take casein, for example, in milk. Okay, milk has got a pH of around 7, pretty neutral. The isoelectric point for casein is 4. 7 is above 4, so that means that casein protein has a net charge of negative. So when you start lowering the pH in milk, like if it's sitting out on a counter fermenting, that bacteria will produce lactic acid. Lactic acid lowers the pH. It lowers it to the point where the pH is now at 4. That's when your milk curdles at pH 4, because that's the isoelectric point of casein. That's also how you make cheese. You put in vinegar, lower the pH value to 4, and your curds come out of your milk. Leaves the whey behind. So isoelectric point is a very important concept. Okay, because amino acids are Zwitter ions, they're very ionic. They have a very high IMF, very, very high, because it's ionic. Hence, high melting point, high surface tension, high boiling point, etc. Again, another real important reaction. This is with, again, the cysteine amino acid. Now, what happens is the SH group can bond with another SH group. Can happen is you can attach one cysteine molecule to another cysteine molecule, and you'll end up with a cysteine molecule. That's that double bonded sulfur here. Now, if that amino acid is in a protein and the protein is folded, that's going to be a covalent crosslink. Very, very strong. It's the strongest crosslink there is with proteins. And of course, in a reductive environment, that double-sided or a double sulfide bond can go back and be oxidized. Now, when people get perms, this is what they're doing, this reaction right here. So basically your hair, as you all know, protein, and all of your hair, bless you, good old hay fever, and your hair is either straight or curly. And the reason why it's straight or curly is because of the crosslinks in the protein. So when you have a perm, it's in two stages. The first stage is to break the crosslink in a reductive environment. So when you expose your hair to a reductive environment, it relaxes. That's because the disulfide bond goes to two thiols here. It relaxes it. Crosslink is gone. And then you put your hair in whatever you want, either straighten it or curl it, and then you reestablish the disulfide bond again, which makes it permanent because it's strong covalent crosslink. And that's also why perms are very stinky, because you're using oxidative chemicals, reductive chemicals. It's they're pretty harsh. Also, it varies depending on which way the strands of the protein are going. So that you can be the one on the left or the one on the right. Okay, so on Tuesday, we talked about the amide bond. That's when you have an acid and an amine together, it forms an amide. Okay, since amino acids contain an amine and an acid, amino acids can go through an amide reaction. So we have acid, amine, forms an amide, or a peptide bond, which is an old world description of that bond. Now, if we took an amino acid like glycine and alanine and linked them together with an amide bond, there's a 50-50 chance who goes first, who goes second. So you can get a mixture of about 50%. However, enzymatically, you'll get 100% of one or the other. Enzymes are magic. We have three distinctions on peptides. Short, medium, and long. The short one is just called a peptide. Ones that are long, but under 50 is called a polypeptide. Anything over 50 is considered a protein. So 3 groupings of polypeptides, and that's just the number of amino acids. I said this earlier, but you always put the amino end at the left side. That's called the terminal end. And the acidic part on the right. I call it C-terminal, N-terminal. To name a polypeptide, okay, you've got the amino acid on the right is the main body. Think of that like a backbone. So this is valine here. The next one in is glycerin. And notice it's like an YL. And then the first one then is alanine. Now this gets very, very, very cumbersome after a while. If you have a polypeptide with 50 amino acids in it, it's a really long name. Now, when we talk about proteins, 50 or more amino acids, they take on the characteristics of the amino acids and the characteristics of the functional groups coming off of the amino acids. There's 20 different versions. So proteins, polypeptides, and peptides all have an isoelectric point. All three. All three also take the form of a Zwitter ion, the end and C terminal ends take on the Zwitter ion. So the N terminal end is always positive. The C terminal end is always negative. That's the part that forms a Zwitter ion. Does anyone speak German? I think Zwitter is a German word. I'm curious. Okay. A buffer. Did you learn about buffers in 305? A little bit. A buffer will maintain a certain pH depending on what the weak acid is that makes up the buffer. A weak acid and the associated base is how you make a buffer. So we've got both of those in the case of a protein, polypeptide, and peptide. And because of that, they have an associated buffer pH. And that's what leads to the isoelectric point. So because amino acids can act as buffers, that's where the isoelectric point is derived. Okay. When you have a solution with a protein in it, all solutions have a pH associated with it. So then you need to look at 2 things, what the pH of the solution is and the isoelectric point of that protein in there. If they're the same, that means that protein has no charge, has no net charge. Hence, if it's in water, like our body, for example, or milk or whatever, then it's going to curdle out, precipitate out. So this relationship between is real important. Isoelectric point, pH. And again, so cheese is made by moving the pH of the solution to the isoelectric point of the protein. Bacteria make lactic acid. They're called lactobacillus. Those lactobacillus bacteria produce lactic acid. Lactic acid lowers the pH down and down and down until it hits 4. And then the curds precipitate out from the cheese, making cheese. Okay, there are 2 major types of proteins, fibrous and globular. Fibrous are typically when you think of meat, think of fibrous proteins. They're very long, they're very sturdy proteins. Globular are the exact opposite. They're not long and skinny, they're globular shaped, like globs, and they're also very delicate. Enzymes are globular proteins. So enzymes will be very, very fragile. Now, the reason why they're globular has to do with the sequence of the amino acids that make up the protein. Depends on their functional groups. Now, if we're in a water environment, like this is water, the functional groups that are polar are going to want to go to the outside and force that protein shape so that the polar ones on the outside, so these are all very polar. The non-polar ones are in the middle. Just like when we had the fats. Remember we had the micelle, we had the non-polar ones in the middle, polar ones on the outside. Same concept. Now, proteins can get quite complicated because of their size. They can also coagulate with other proteins to form a conjugated protein. And that includes also things like metals, not necessarily other proteins. So you can combine a protein with another protein and or another group like this. Now this is heme. This is what hemoglobin is based on. And there's the iron in the middle. It's like iron carries oxygen really well. In fact, they were talking about that in the 401 class before us today. Okay. Now there's lots of different kinds of proteins. Lots. I've seen estimates of 1 million or so different kinds of proteins, just a whole bunch of them. And they've kind of classed them into these 6 basic groupings. There's a nucleoprotein, like DNA, RNA, mRNA, tRNA, et cetera. Lipoproteins, they can hook up with lipids. Glycoproteins, they can hook up with carbohydrates. Phosphoproteins, lots of phosphate groups in there. Hemoproteins, these are all iron-based. And then metalloproteins, where they hook up with some kind of a heavy metal. Now, the other thing is you can have a lipoglycoprotein as well. So if these, for example, hook up together, there are all those combinations as well. That's why there's so many protein combinations out there. Now, there are 4 levels of structural organization in proteins. First one is primary. That's just a sequence of amino acids. That's all it is. It looks like a long train. Sequence of amino acids. Okay. Second thing is secondary. And then tertiary. Now those have to do with how the protein is folded. Okay, so the secondary has to do with a couple things. This is hydrogen bonding. So as that protein on the functional groups fold and get near one another, then the functional groups start either attracting or repulsing. If they're attracting, that means that forms a cross-link. There is two major ways proteins fold, alpha and beta. They're called alpha helix, which looks like the traditional DNA molecule, and a pleated sheet. Most proteins are a combination of the two. Not one is not, except for DNA, that's pretty pure, but usually there are combinations of these together. Okay, so let's look at this now. The primary structure on this particular protein is here. That's the sequence of the amino acids. Here is where you get that hydrogen bonding attraction going on. And that's what links this up and makes it twist. Those are examples of a pure alpha helix. Also, I want to add the other way is this pleated sheet idea. And basically that's strands of amino acids lying adjacent to one another. And mention up there, silk looks like this microscopically. And silk is the only pleated sheet protein out there. Pleated sheets are very common. They're just in combination with alpha helix. Okay, so this is a nice example of a typical protein. So you've got an alpha helix, pleated sheet, and then kind of a generic folding. Most all the proteins look like this, with a combination of alpha and beta structures. Now, there are four kinds of crosslinks that keep all these together. The strongest one is this one. I'm going to give it a value of 1 for being the strongest. Okay? Because when you look at intermolecular forces, ionic is at the top, but even higher up on that is covalent. And they don't consider covalent as intermolecular. Ionic is, though. Okay, so here's ionic here. Remember on our list of intermolecular forces, ionic is at the top. So if you have an ionic crosslink, that's going to be pretty strong. Here we've got hydrogen bonding. And then this is just LDF. So what's the weakest? This is important because this determines the functionality of the protein. Is this protein going to be an amino acid? Excuse me, an enzyme. It's going to be an enzyme. Then all these are going to interact to form something called an active site. And that's where all the magic happens with an enzyme. So these functional groups on the alpha carbon on those amino acids determine the shape of the protein, okay? So we have the functional amino acid groups determine the structure and how those functional groups interact with one another through our different kinds of IMFs, including covalent. So this is important, this here. Okay, like our fats forming micelles in a water environment, which our body is, the functional groups on the amino acids are going to tend to want to be on the outside, so they interact with the polar water. The non-polar functional groups want to be on the inside, as far away from the polar water as possible. So exactly the same reason a micelle forms with fats. This interaction is exactly the same thing. And then the most complicated organizational structure for proteins is called quaternary. And that quaternary one involves subunits. The subunits can be other protein or a group like this, and they call those prosthetic groups. Just like a prosthetic limb or leg, it's an add-on. All of you have heard of hemoglobin. This is what it looks like. So it's a very large protein. It has 4 units, 4 other proteins in it. And in the middle, and this is where the iron prosthetic group resides, in that hemoglobin molecule. Okay, so going back to the chemistry of amides, they're susceptible in acidic environments and basic environments. They will hydrolyze, they will come apart. So the principal mechanism for taking a protein into its subsequent amino acids is through hydrolysis, and that happens in our stomach. And the enzyme is called a protease. And the protease to amino acids. Now, some proteases that are designed, for example, in our stomach, are designed to be in an acidic environment. Your pH of your stomach is 2-ish. So if the enzyme doesn't have a pH of 2, it doesn't work very well because the shape will change depending on pH. So protein shape is determined by temperature, pH. Temperature and pH. Think about when you cook. Here you have a heat. Think about when you make ceviche. Has anyone ever eaten ceviche in here? Okay, you know how to make it? Very easy. You take and basically add a really strong vinaigrette to the shrimp or scallops or whatever you're done. And what happens is that pH will cook the meat. at room temperature, because it's meat cooked is the protein changes shape. So if you can change the shape at room temperature, you have, quote unquote, cooked the meat by subjecting it to a much lower pH than it's designed for. Same with heating. Heating will change the shape of a protein as well. Also, it depends on the solvent the protein is in. So all those are factors that determine the shape. So, and typically when proteins are hydrolyzed, they go in steps, smaller and smaller and smaller, till eventually they get onto amino acids. So, protein denaturization is a fancy way of saying the shape of the protein is now different from when it first was formed. There's permanent and semi-permanent denaturization. When it's permanent, it can't go back. You take an egg, you fry it, it cannot go back to the original gooey egg. It's a one-way trip because the temperature is high. So we have heat. Now this is situations that will denature a protein. Okay, heat / ultraviolet light. So when you go out in the ultraviolet light in the sun, that sun denatures the protein on your skin. When we change the solvent that the protein is in, its shape will change. When you're in an anatomy physiology class, you look at a lot of flesh, a lot of tissue, and it's all in formaldehyde. They call it formalin, which is diluted formaldehyde. Formaldehyde changes the protein. It denatures the protein that is in the solution. When you're embalmed, they inject you with formalin, same stuff. Okay. Strong acids or bases. Just like when we do ceviche, we're introducing a pH change by acid. Now, detergents and soaps are emulsifiers. Emulsifiers, as you remember, have a polar end and a non-polar end. So what happens is it can get in there and start interacting with the side chains on the amino acid in the protein. And it'll form crosslinks that are not supposed to be there. Hence, it'll change the shape of the protein. And these guys, heavy metals, forms with the cysteine amino acid. If there's a cysteine amino acid in the chain of the protein and it's exposed to a heavy metal, then that is going to change the shape of that protein. If that protein is an enzyme, the enzyme will no longer work. And this is usually very permanent. So you can't fix it. Okay, so that's proteins. Proteins are pretty cool. One of the ways of purifying a protein, say we have two proteins and they have different isoelectric values. So what you can do is a very common technique in biochemistry, purify proteins, is you change the pH of the solution the two proteins are in. And when you approach the isoelectric point of one of them, it'll precipitate out. Then you stop adding acid or base, and then you filter it. And then what's in the filter paper is going to be the protein that you've reached the isoelectric point on. And the other one's still in solution. It's a really handy way to purify proteins. That would be a good lab for us to do, but may have to invent one. Okay, so that's basically it on proteins. Now, that's our subunit. What I'd like to do on Tuesday is review reactions. I'm going to go over one of the old tests, practice tests, and talk about the reactions. Now, the hard part on this unit is going to be the well, knowing the amino acids, number one, but also it's the same old stuff. It's reactions. And then amides are tricky to name because of the if there's a group hanging off of the nitrogen, you've got to do the N in the name. So there's some trickier parts to this unit in terms of naming. So anyway, we're going to practice that on Tuesday. Okay. It's going to get cold this weekend. Highs 54, as opposed to today, highs 85.